So guys, the first factor which affects the rate of reaction is the substrate concentration. Now when you plot the velocity and substrate concentration, right, you can see the curve like this. So what is happening? Initially the curve is straight line, right? Means the enzymes active sites are now binding the substrate, isn't it? So when they are binding the substrate and the product is formed, right? And the enzymes active sites are not saturated till now. So more the enzyme, more the substrate, and they keep on increasing the product concentration, right? So velocity is increasing in the linear fashion. Fine. So that is our first order kinetics, right? Because right now the active site of the enzyme is not saturated. There are still free enzymes which are able to bind the substrate. Okay. Now when we are increasing the substrate concentration, as you can see, there is a hyperbolic curve, right? So guys, this hyperbolic curve simply means it's a zero order kinetics. That means now the active site of enzymes are completely saturated with substrate, right? Suppose I had 100 enzymes and now I have 150 substrates. So 50, all the 100 substrates are already bound to the active site. So this may cause the plateau or the hyperbolic curve. So that is zero order kinetics. That means now the velocity or the rate of reaction is independent of substrate concentration, right guys? So this is the curve, as you can see here, that is a curve of substrate concentration which was studied by Michaelis and Menten, right? So Michaelis and Menten found that when the substrate concentration is low, right, the enzyme's velocity or velocity of the reaction is directly proportional to substrate concentration. When the substrate concentration is very high, then the rate of reaction or the velocity of reaction is independent. It will become maximum, and now it is independent of the substrate concentration. So this point is called as Vmax. What is Vmax? The maximum velocity achieved by the enzyme during the reaction, right? Okay. So this is a point that is called as Vmax, where all the enzymes are already bound to the substrate. No free enzyme left. So velocity becomes now constant, almost, right? Now, guys, as you can see in the curve, there is also one term mentioned on the x-axis, and that is the substrate axis, that is Km. So this is called as Michaelis-Menten's constant. So what is Michaelis-Menten's constant? Guys, this is the substrate concentration at which Vmax by 2 is achieved. So don't say Km is velocity. Km is substrate, as we are mentioning already on the x-axis, right? Substrate axis. So this is the substrate concentration at which Vmax by 2 is achieved, right?